Okay, let's unpack this. If you were trying to map out a massive, bustling metropolis,
Yeah,
you wouldn't just look at the asphalt on the roads.
right. No, that wouldn't give you the whole picture at all.
Exactly. I mean, you would need a schematic of the subway lines, the electrical grid, the water pipes, the foot traffic patterns.
Yeah, all the different layers that make the city actually function.
Right, because if you only look at one of those layers, you are going to fundamentally miss the real story of how the city works. And, well, that is exactly the friction we face when trying to map the human brain.
Oh, absolutely.
Oh.
Especially when you're hunting for complex, cascading neurodegenerative diseases, like Alzheimer's.
Yeah, which brings us to today. Welcome to this deep dive. Today we have a genuinely fascinating research paper. It's from May 2026, and it's titled NeuroAgent, LLM Agents for Multimodal Neuroimaging Analysis and Research.
It's quite a read, honestly.
It really is. And our mission for you today is to explore how artificial intelligence has evolved. Because, I mean, we are not talking about a simple chatbot that helps you draft emails anymore.
No, not at all.
Mhm.
We are looking at an AI acting as a full-fledged autonomous project manager.
Yeah, a tireless lab assistant that, writes its own code, debugs its own errors, and untangles the absolute messiest data in science-human brain scans.
The shift in capability here, I mean, it really cannot be overstated. Historically, combining all those different maps of the brain has just been an operational nightmare.
Right.
As anyone who has worked in neuroimaging knows, the temporal and spatial resolutions of different scan types, they sort of fight against each other.
Yeah, it's a constant battle, isn't it?
It is. But it is a necessary friction, though, because brain diseases operate on multiple dimensions simultaneously. You have to see all of it.
So before we get to the AI architecture that attempts to solve this, I feel like we need to understand the severity of the human bottleneck it's replacing.
Definitely.
Because when we look at section 1 of the paper, the authors don't just list the modalities involved in modern Alzheimer's research. They really outline the profound friction of integrating them.
They do, yeah.
Like, we all know you need structural MRI for the macroscopic anatomy and functional MRI for the resting state network connectivity, and then DTI for the microscopic white matter tracks.
Right, and PET scans for molecular protein readouts, like tau.
Exactly. But forcing those files to communicate, I mean, that is where the science just grinds to a halt.
It creates a severe bottleneck that, honestly, ultimately compromises the scientific method itself. The inconsistency in how humans handle this data is staggering.
Oh, yeah, the researchers highlight this brilliantly.
Right.
Before we get into it, I just have to mention, in section 2.1, they reference the RPS study.
Ah, yes. The Neuroimaging Analysis, Representation, and Prediction Study. For those who might not recall, the premise of NeuroPS was surprisingly simple.
Tell us about it.
Well, they gave 70 different independent research teams the exact same fMRI dataset.
Okay, the exact same raw data.
Exactly. And they asked them to test the exact same 9 hypotheses.
I mean, in a perfect scientific world, that should yield 70 identical, or at least functionally identical conclusions, right?
You would certainly hope so, yeah. But because of manual differences in how those teams pre-processed the data, like the specific spatial smoothing parameters they chose or varying statistical thresholds.
Let me guess. Different software packages.
Exactly. Because of all that, those 70 teams came to completely divergent conclusions regarding the hypotheses. Out of 70 teams, there were 0 identical analysis workflows.
0. Wow, I mean, it's the equivalent of trying to bake a single uniform cake using 4 different recipes written in 4 different languages.
That's a great way to put it.
And if you stir the batter just slightly differently than the guy at the bench next to you, you pull a totally different cake out of the oven.
That is an excellent analogy. Now, I want you, the person listening right now, to put yourself in the shoes of a highly trained computational neuroscientist.
Okay, I'm wearing the lab coat.
Right. So your life's goal is to uncover the biological mechanisms of Alzheimer's disease. But in reality, you are spending 80% of your working hours manually formatting files.
The busy work.
Exactly. Writing fragile bash scripts, tweaking spatial registration parameters, converting data types, just so your DTI scan can physically overlay onto your MRI scan without crashing the software.
It's just an immense drain on specialized human capital, which I guess brings us to the proposed solution. NeuroAgent.
Yes, the multi-agent framework.
So how does this actually remove that human bottleneck? Because, automating file conversions is one thing, but orchestrating a whole multimodal scientific pipeline, that's something entirely different.
It is. The architecture is what really sets this apart from a standard automation script.
Okay, so let's get into the architecture. In Figure 1 in Section 3.1 of the paper, the authors break down this hierarchical multi-agent framework.
Right, so at the very top of the hierarchy is what they call the central orchestrator.
The big boss.
Essentially, yeah.
Yeah.
This is the primary LLM-based planner. It takes a natural language request from the user, parses the scientific intent, determines exactly which imaging modalities are required, and then maps out a step-by-step workflow.
And then what?
Once the plan is set, it delegates the specific tasks down to the specialized modality agents. So there is one dedicated agent for structural MRI, another isolated agent for fMRI, another for DTI, and so forth.
Okay, so if we move away from the kitchen analogies, it sounds a lot like a general contractor building a custom house.
I like that, yeah.
Like the central orchestrator is the general contractor looking at the master blueprint. They don't do the plumbing themselves. They sequence the timeline and, yell out the orders to the specialized subcontractors.
Right, like the framing agent needs to build the walls before the electrical agent can run the wiring.
Exactly. And the plumbing agent needs to snake the pipes without crossing the electrical lines.
That captures the hierarchy perfectly. And those specialized agents are isolated so they can focus entirely on the unique computational physics of their specific modality.
Makes sense. But what happens when things go wrong? Like traditional rigid coding pipelines fail the moment a file format is unexpected.
Oh, absolutely. But here's the real breakthrough. According to Section 3.1.2, NeuroAgent utilizes what they call a generate execute validate engine.
Okay, break that down for us.
The AI dynamically writes custom Python code for the specific task at hand. It executes it within a secure, isolated sandbox, and then it evaluates the outcome.
And if it crashes, like let's say a nonlinear brain registration tool diverges because, I don't know, the skull stripping failed.
The agent does not freeze and wait for human intervention.
Wait, really? It just keeps going?
Yes. It captures the Python error traceback log, feeds that error back into its own context window, deduces why the logic failed, rewrites the code, and executes it again.
I have to pause there. Because, the idea of an AI catching its own traceback errors and rewriting its own logic is impressive, but it raises an immediate red flag.
I know exactly what you're going to say.
Right, like what prevents the agent from getting trapped in an infinite loop of bad code? Or worse, what if the code executes perfectly without errors, but the resulting scientific output is just, statistical garbage?
That is the most critical vulnerability in autonomous science, period. And the authors anticipated it. This is why NeuroAgent is not a completely unsupervised black box.
Okay, so there's a safety net.
Yes. The paper details a strict human-in-the-loop fail-safe mechanism. The system is constrained by rigid scientific schemas.
Ah, okay.
It validates the structural integrity of the output at every single step against these schemas. If the agent hits a wall, it cannot self-correct within a set number of iterations, or if the output data distribution looks anomalous compared to expected biological baselines.
It stops.
Exactly. It halts and escalates the specific problem to a human researcher with a detailed diagnostic report.
So the human remains the ultimate authority.
Precisely. But they're only stepping in to resolve complex edge cases, rather than babysitting every single routine file conversion.
Okay, the theoretical framework is undeniably elegant. You have a general contractor managing a team of self-correcting specialists, all backed by a human safety net.
It's very well designed.
But, I am always skeptical of architectural theory until I see the benchmarks. When they actually tested these agents, how intelligent were they in practice?
Well, the researchers ran extensive pipeline ablation studies to benchmark different large language models across the various operational stages, and the results are genuinely counterintuitive.
Really? How so?
Well, if we examine Table 4, which outlines the benchmarking for the intent parsing stage.
That's how accurately the AI translates the initial natural language prompt into the right sequence of tools, right?
Exactly. We see a fascinating divergence there. They tested several models within the same family, and a highly compact 𝗀3/4b model, which only has 4 billion parameters.
Just 4 billion? That's tiny for an LLM.
I know. But it achieved a 100% accuracy rate on joint exact matches.
Wait, 100%?
Yes. But here is the kicker. The intermediate models, specifically the 14 billion and 8 billion parameter models, failed entirely. They returned a 100% invalid rate.
Wait, I have to challenge that. Everything we know about the current trajectory of large language models dictates that parameter count is king. The larger the model, the better the reasoning capabilities.
Generally, yes.
So how on earth does a 4 billion parameter model completely outsmart a 14 billion parameter model on a complex logical mapping task?
It comes down to the strict operational constraints of the task itself.
Yeah.
In medical orchestration, the output cannot just be correct in its reasoning.
Okay.
It must be perfectly formatted as structured JSON data so the downstream software can parse it. And larger models are heavily fine-tuned to be highly conversational and helpful.
Oh, I see where this is going.
Yeah, consequently, they have a tendency to overthink and produce what we call chatty responses. The 14 billion parameter model would deduce the correct sequence of tools, but it would wrap that JSON output in a conversational paragraph.
Saying something like, here is the workflow you requested based on the modality.
Exactly. And that extra text completely breaks the downstream code parser.
Oh.
The smaller model, lacking that conversational overhead, simply adhered to the strict JSON formatting rules with robotic precision.
That is fascinating.
It really demonstrates that in scientific workflow automation, strict compliance and formatting discipline are often more valuable than sheer computational size.
That makes a lot of sense. You don't want your electrician giving you a 10-minute monologue about the history of copper wiring.
No, definitely not.
You just want them to wire the house according to the schematic. But the AI cannot be flawless across the entire pipeline. Where do these specialized agents still hit a wall?
Oh, they absolutely still struggle, and the authors are very transparent about it.
Yep.
When you look at Table 5 and Table 6, you see exactly where the pipeline chokes.
And where is that?
It is overwhelmingly in data integration. While the agents excel at writing Python code to process a single fMRI scan, assembling a massive, multi-subject clinical manifest from disparate sources remains a profound challenge.
Really? How bad is it?
Across the various models tested, the all-pass rate for the data integration tasks hovered between a mere 37.5 and 50%.
Wait, why is data integration so uniquely difficult for an LLM? If it can write a complex spatial registration algorithm, why does it fail at putting files in a folder?
Because real-world clinical data is notoriously messy and disorganized. In theory, neuroimaging data should be formatted according to strict standards, like BIDS the brain imaging data structure.
Right, but in reality?
In reality, DICOM headers are full of typos. A patient might have their structural MRI on a Tuesday, but the machine broke, so their PT scan was done on Thursday.
Oh, I see.
And the file naming conventions might vary slightly between the 2 days. To an LLM, matching disparate subject IDs and reconciling those temporal misalignments requires a level of strict relational database logic that language models fundamentally lack.
They try to find semantic patterns where they should be using rigid relational joins.
Exactly. That integration phase is where the human-in-the-loop mechanism is activated most frequently.
Okay, that highlights the gap between simulated benchmarks and the messy reality of a hospital server. But, let's push past the data organization. Let's talk about the actual science.
Let's do it.
Writing code and formatting JSON is great for saving time, but can this autonomous system actually produce valid clinical discoveries?
It can. And the way it does it is what makes this research so compelling.
Okay, I'm ready.
According to Section 4.4, and visualized in Figure 2 and Figure 3, the researchers demonstrated a complete end-to-end cortical thickness analysis.
So measuring how the brain's outer layer changes.
Exactly. And the human user did not write a single line of statistical modeling code. They simply provided a natural language prompt instructing the agent to analyze cortical thinning across 3 groups.
Which groups?
Cognitively normal subjects, those with mild cognitive impairment, and patients with Alzheimer's disease.
Here's where it gets really interesting.
It really does. The agent independently queried the database, retrieved the appropriate structural MRIs, wrote the preprocessing scripts, and then executed the statistical regressions.
And did it adjust for anything, like age?
Yes, crucially, automatically adjusting for confounding variables like patient age and sex.
Wow.
And the agent correctly identified a statistically significant pattern of Alzheimer's-related cortical thinning, specifically localized in the entorhinal cortex.
That's a known biomarker, right?
Yes. It even independently generated publication-ready box plots and scatter plots, visualizing this stepwise reduction in thickness as the disease progressed.
Okay. It is impressive, but my skepticism is flaring up again.
Fair enough.
If an AI is autonomously querying data and running regressions until it finds a result, how do we know it isn't just p-hacking? I mean, it's very easy to run thousands of statistical permutations on a massive data set until you find one random correlation that looks like a biomarker on a scatter plot.
That is exactly why the predefined schemas we talked about are so critical. The agent isn't allowed to arbitrarily fish through the data. Its statistical workflow is strictly constrained by standard neuroimaging pipelines.
So it knew the right way to look.
Exactly. It didn't discover the entorhinal cortex anomaly by random chance. It successfully replicated one of the most well-established anatomical biomarkers of Alzheimer's disease using a standardized workflow.
Okay, that's reassuring.
And it didn't stop at structural MRI. Referencing Figure 4 and Figure 5, the authors detail how the system applied the same natural language workflow to diffusion MRI, the DTI data.
Right, the microscopic white matter wiring.
Precisely. Without hand-holding, the agent correctly identified age-related structural trajectories in the inferior parietal cortex.
What exactly did it find?
It autonomously calculated that as patients age, there is a measurable decline in fractional anisotropy coupled with an increase in mean diffusivity.
Okay, in clinical terms, what does that mean?
It means the protective myelin around the microscopic white matter tracts is degrading, allowing water molecules to diffuse more randomly.
Oh, wow. And the system successfully extracted these complex, multi-layered clinical biomarkers entirely on its own.
Entirely on its own.
I mean, a researcher no longer needs to possess a PhD in computer science just to ask a biological question. They can literally converse with the data.
It democratizes the analysis in a huge way.
But, we've established it can process group trends and identify known biological markers. But the ultimate crucible for any medical technology is the individual patient diagnosis.
That's the holy grail.
Right. Group averages are one thing, but Can this system synthesize all of this messy, multimodal data to accurately predict if a specific individual patient has Alzheimer's?
Well, this is the climax of the paper's findings. In Section 4.5 in Able 10, the researchers subjected NeuroAgent to the ultimate diagnostic challenge.
Okay, lay it on me.
They took all the disparate modalities the system had autonomously pre-processed, the structural MRIs, the functional MRIs, the molecular tau PET scans, and the tabular clinical data like age and cognitive scores.
The whole map of the city.
The whole map. They then fused this heterogeneous data together using what they call an agent ensemble. And the resulting diagnostic model achieved an area under the curve, or AUC, of 0.9518.
Okay, for those who don't spend their days reading statistical methodology, an AUC is a measure of diagnostic accuracy. An AUC of 1.0 represents a flawless, perfect test. An AUC of 0.5 is no better than flipping a coin.
Exactly.
So hitting an AUC of over 0.95 for a disease as insidious and complex as Alzheimer's is an incredibly high watermark.
It is a remarkable achievement. But to truly appreciate the power of the agent ensemble, you have to understand how it synthesizes the data and compare it to the single-modality baselines.
How does it work?
The ensemble uses a late fusion mechanism. It doesn't just match all the pixels together. Each modality agent extracts its own high-level diagnostic features, and then the central orchestrator weighs those features against each other to make a final prediction.
And what were the baselines?
When we look at Table 7 and Table 8, the researchers tested standard deep learning architectures, like Resonant 18, on isolated modalities. If you ask a standard model to diagnose the patient using only their structural MRI, the AUC drops significantly to roughly 0.86.
Okay, and the PT scans?
If you rely solely on the tau PET scan, it improves slightly to about 0.90.
Oh, I see.
The 95% accuracy is only unlocked when the AI seamlessly orchestrates and mathematically fuses all four modalities together.
It goes back to the city map. The AI can finally see the subway, the power grid, and the traffic patterns all at once, and it understands how a failure in one system cascades into the others.
Exactly. It's the synthesis that makes the difference.
So what does this all mean for the listener? I mean, maybe you aren't a clinical neuroscientist writing grant proposals. Why should the integration of an LLM orchestrator matter to you?
It matters fundamentally because of what it means for the velocity of medical innovation.
How so?
Consider the logistics of a massive clinical trial for a new Alzheimer's therapeutic. Those trials require analyzing thousands of these multimodal scans across hundreds of clinical sites.
Right, mountains of data.
Exactly. And if highly trained researchers are spending 80% of their time manually standardizing bias formats and debugging spatial registration errors, those trials take years longer to complete.
And they cost tens of millions of dollars more, I'm sure.
Without a doubt. By deploying a system that achieves a 95% accuracy rate using fully automated self-correcting preparation, we can execute complex clinical trials at a fraction of the historical time and cost.
It's just a massive efficiency boost.
It is a critical infrastructure upgrade that dramatically accelerates our path toward individualized precision medicine.
We are transitioning from researchers acting as manual data janitors to researchers acting as directors of brilliant AI orchestrators. Let's take a moment to recap this entire journey because the implications are just vast.
They really are.
We started with the human bottleneck, right? The nightmare of multimodal brain scans where 70 different expert teams couldn't even agree on the same raw data set due to pipeline variation.
The NRPS study, yeah.
And then we explored the NeuroAgent architecture, a central orchestrator acting as a general contractor, utilizing a generate-execute-validate loop to literally write its own Python code, read its own error tracebacks, and fix its own bugs.
All while keeping the human in the loop for the really tricky stuff.
Right. And despite struggling with the messy reality of clinical data integration, this system was able to fuse 4 different types of neuroimaging data to achieve a staggering 95% diagnostic accuracy for Alzheimer's disease.
It's incredible.
And all of this was initiated by a scientist simply typing a request in plain English. It is a fundamental paradigm shift in how scientific research will be conducted.
The paradigm shift is undeniably here. And, well, it raises an unavoidable question, something for you to contemplate long after this deep dive concludes.
I love a good lingering thought. What is it?
Right now, we are rightfully marveling at the fact that an AI framework can autonomously design the computational workflow, debug the syntax errors, and extract the established biological markers we ask it to find.
Right.
But as these models scale their reasoning capabilities, I mean, how long until the AI is no longer just processing the multimodal data we give it, but actually formulating completely novel, counterintuitive scientific hypotheses about the brain that human doctors never even thought to ask?
Oh, wow. Yeah, if AI can finally see the entire city grid at once, it might just discover pathways and connections we never knew existed.
Exactly.
Well, thanks for joining us on this deep dive. We'll catch you next time.